remember, red blood cells have no nucleus, or as we say, they are enucleated. Okay, that is important to increase surface area for transport of oxygen. Okay, there is no mitochondria because you do not want the red blood cell to be using up the oxygen for respiration during transport. You want the red blood cells to deliver all the oxygen that it gets from the lungs, to the cells of the body. Okay, so that is important. So it's important to note that red blood cells are not capable of cellular respiration, right? They also have a biconcave surface, okay, that gives them a larger surface area, and even larger surface area, okay, for oxygen to be transported, okay. The cytoplasm also has a large amount of hemoglobin, okay, that gives it its red color, okay. Each hemoglobin molecule also is able to bind with many oxygen molecules, okay, so that these oxygen molecules can be transported around the body. We'll come to hemoglobin in a while. Okay, good. So the next component of blood is actually a white blood cell. It's phagocytes. Okay, phago means to eat. Okay, so they fight infections by engulfing and digesting those foreign elements. So the first thing that would happen is that the phagocyte in the host cell would bind or connect or form a connection with the foreign invading cell. Okay, this is an invader. This is an invader in the form of a virus. So the phagocyte would recognize the virus as foreign or invasive. It would bind to it, okay. So the cell would bind to the foreign cell, and that would then the membrane of the phagocyte would start to fold inwards, okay. And as it folds inwards, it surrounds the virus. So that is what is happening here. The host cell is surrounding the virus. Okay, let me just write that. Okay, host cell T. The host cell surrounds the virus. All right. And it would continue doing that until the virus is completely surrounded. Okay, it would continue to do that until the virus is completely surrounded. Okay, when the virus is completely surrounded, you get what is called a phagosome being formed on the inside of the host cell. Okay, with the virus in the middle of it. And this, the cell would then secrete enzymes to destroy the virus on the inside. Okay, so host cell, okay, would then secrete enzymes okay, secrete enzymes to destroy virus. Oops. And that's how phagocytes works. Okay, they secrete enzyme, and well, before they secrete enzyme, they engulf. Okay, they engulf the
and I don't understand.
Okay.
It's not working. Wait.
Oh, it don't working.
What's not working?
So you don't understand.
Okay, so the phagocytes, okay, phago means to eat, okay? They fight against foreign invading elements by surrounding those elements, okay, and secreting enzymes to destroy them on the inside, okay? So what would happen is that the host cell would identify the virus, for instance, as an invader. It would bind to the virus, then surround the virus. When it completely surrounds the virus, it would, okay. When it completely surrounds the virus, it would digest the virus on the inside, okay, by secreting enzymes. So the enzyme secreted by the host cell or the phagocyte would digest the virus on the inside, okay. So it secrete enzymes, okay, to digest virus. Okay, sorry. All right, for some reason my pen, all right, to digest virus, okay. And that is how the phagocytes fight against viral infection, okay. So first it must recognize the virus as an invader. That's the first step, recognition. After recognition, it would bind, okay. It would bind to the virus. The host cell would bind to the virus. Then it would start surrounding the virus, okay. When it surrounds the virus completely, it would secrete enzymes to destroy the virus on the inside, okay. Okay, and that act of surrounding the virus completely is called, okay, we say it engulfs, ENGUL, okay, engulfs. So we say the host cell engulfs the virus, okay. The host cell engulfs the virus, okay. And that is how phagocytes fight against infection. We also have lymphocytes. Notice the shape. Let's go, if you go back to the phagocyte, look at the diagram to the left here. All these here, this is a nucleus. This is a lobe of the nucleus. This is another lobe. Okay, lobe, L-O-B-E. So the nucleus of a phagocyte has many lobes, okay, of its nucleus. Okay, so that's a key factor in identifying a phagocyte, the multi-lobed nucleus, right? The multi-lobed nucleus, okay, many lobes of its nucleus. So whenever you see that, you know that it's a phagocyte. So you have one lobe here, one here, you have another one here, okay, and so forth. One here, 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 okay, many lobes of the nucleus. The red blood cell, remember the red blood cell has no nucleus, right? No nucleus. So we go from no nucleus to multiple lobes of the nucleus in the phagocyte. And then the lymphocytes, okay, they have one large nucleus, okay. Almost the entire cell is comprised of the nucleus. And there are different types of lymphocytes. So in the diagram to the left here, we have what we call, we have T lymphocytes. Okay, we have T lymphocytes. Okay, T lymphocytes. And we have B lymphocytes. Okay, good. T and B lymphocytes. Okay. CYTES. So you have T and B lymphocytes, and the T lymphocytes would comprise T cells. So you have the cytotoxic T cells. Okay, this would act to destroy cells that are infected with a virus or cells that are damaged over time. Okay, you call them cytotoxic T cells. Okay, cyto means cells, toxic to destroy. Okay, then you have the helper T cells. Okay, right, which helps, as the name suggests, they help these cytotoxic T cells, and they also help the B cells. Now, for your level, we will be focusing more on the B cells. Okay, the B cells are responsible for producing antibodies. Okay, so they produce antibodies to fight infection, or the antibodies would bind to what you call an antigen. Okay, so let me write it here: antibodies. Oops. Okay, so the antibodies. Now, every foreign, right, cell or element has different markers on the surface. Okay, so let's say, one moment, let me just share a whiteboard. Okay, let me share the whiteboard. Right, everyone's seen the whiteboard, right? So here we have a cell. Okay, on the surface of this cell you have markers. Okay, or you could call them identifiers if you wish, and this helps the body's immune system recognize those cells as either domestic cells or foreign cells. Now you have these, and you have antibodies. So the B cells, okay, the B cells or the B lymphocytes, they would produce antibodies. Okay, but generally what happens, each cell in the body have those antigens. Okay, those antigens, these are antigens. ANTIGENS. So these are antigens. Okay, okay, they are cell surface markers. Cell surface okay markers. So they help identify the cells. So in every person, every cell has those markers or those identifiers. So your body's immune system would recognize that these are your cells because it has a marker that the cells of the immune system could identify as not foreign. However, if an invading cell steps in, okay, if an invading cell steps in, the B cells of the immune system would secrete antibodies, okay, will secrete antibodies that would destroy those cells. Antibodies, okay. Antibodies are special types of proteins that are functional in the immune system. So they secrete antibodies. The antibodies would bind to antigens, okay? They would bind to antigens or foreign antigens, okay? Foreign antigens. Okay. And when they bind to the antigens, the antibodies would also bind to each other. Okay. They would bind to other antibodies. Bind to other antibodies. So we say when they bind to other antibodies, so after, but they must bind to the antigens first. Okay, so they bind to the antigens first, then the antigen antibody complex binds to the other antigen antibody complexes, and they form a clump. Okay, they form a clump, and that clump would be so compact that the phagocytes would now come and surround them and digest them on the inside. So let me just clear all this so I could draw one diagram. So this, let's say this is a cell and this is an antigen. You have an antibody. Antibodies are Y-shaped. They shape like a Y. Okay, so this is one antigen antibody complex. Okay, then you may have another foreign cell from the same organism here. Okay, then you have this antibody binding to it. The antibody here would bind to this antibody here. Okay, and let's say you have another one here. Let me just bring up that. Let's say you have another cell here with a antigen and the antibody binds. So all three would bind together to form a clump. So this whole thing here is a clump. Okay. This clump is formed okay, and it is eaten by the phagocytes. Okay, I will post this video as usual on Edmodo, so make sure when you're looking at it you go over that again, because it's very important that you understand what is going on here. Okay, and you would also need to read the part in the textbook that speaks on that. Okay, so the antibody, first the immune system recognize the antigens that are foreign or that are not local to the organism or to the cell. Then the B cells would produce antibodies that is able to bind to those antigens. So if normally, if you have an antigen on the surface of your cells called antigen A, your bodies would not produce antibodies that would, or your B cells would not produce antibodies that's able to bind to antigen A. Because if the body does that, then it would be destroying its own cells. Okay, and in that case, you would have what you call an autoimmune system. Well, an autoimmune response, not system, where the body's immune system destroys its own cells. Okay, and something like that happens in a disease called lupus. Okay, so lupus is an example of an autoimmune response where the body's immune system fight against its own cells. Okay, but that should not happen. So the body would produce antibodies that cannot bind to the antigens that it has. Okay, but if a foreign cell comes in, the body would produce antibodies that is able to bind to the antigens on the foreign cell. Okay, after the binding of antibody and antigen, you get an antibody antigen complex. Okay, and that antibody antigen complex would bind to other antibody antigen complexes, and they would form a clump. Then the phagocyte would come and engulf or surround that clump. Okay, so let's say this is the phagocyte here. It would surround the clump. Okay, and after it's completely surrounded, it would secrete enzyme to destroy the clump on the inside. And that is how your body fights off infections. Okay, good. So let me stop share this and go back and share the previous screen. Okay, one moment. Good. So this is a function of the B cells. B cells produce antibodies that fight against antigens. Okay, antibodies fight against antigens. Good. Now, let's look at the function of red blood cells. Red blood cells' main function is to transport oxygen from the lungs to the cells of the body, where they are needed for cellular respiration. The red blood cell contains hemoglobin, okay? And the hemoglobin is a red pigment. It's actually what gives the blood its red color, okay? This hemoglobin has 4 iron atoms within its structure. You would see a structure of hemoglobin in a while. It has 4 iron atoms in its structure, okay? And each of those iron atoms can bind to 4 oxygen molecules, okay? So one hemoglobin, not each iron, sorry, they can bind with 1 oxygen molecule. One hemoglobin molecule can bind reversibly with 4 molecules of oxygen at a time, okay? So that's why they have the 4 iron molecules, so that they would be able to bind with 4 oxygen to form iron oxide. The bond between the iron and oxygen of hemoglobin, the oxygen and the iron of hemoglobin is not permanent. So we say the the hemoglobin binds reversibly with oxygen. In the case of carbon monoxide, okay, carbon monoxide binds irreversibly, okay, and that's where the problem lies. So if you have carbon, okay, carbon monoxide, that's carbon and one oxygen, mono means one. Carbon monoxide binds irreversibly with hemoglobin, which means that once it binds, the bond is permanent. Okay, once it binds, the bond is permanent. It binds irreversibly. Okay, with hemoglobin. Okay, so it binds irreversibly with hemoglobin. And in doing that, it prevents oxygen from binding. Okay, so then the result is that eventually the cells of the body and the tissues and so forth would not get enough oxygen that they need to get, and then the person would suffocate. And if they suffocate for long enough, they would die. Okay, now once the oxygen binds to hemoglobin, the blood becomes bright red because of the formation of oxyhemoglobin. Okay, so when oxygen binds to hemoglobin, you get this complex called oxyhemoglobin. Yes, Hope? Hope? I'm not hearing you, Hope. Your mic is muted. Okay, so when the oxygen binds with hemoglobin, a complex is formed, and that complex is called oxyhemoglobin. When the oxygen is released at the site of the cells, you get deoxyhemoglobin forming. Okay, that's hemoglobin without oxygen. So know the difference between oxyhemoglobin and the deoxyhemoglobin. Okay, so one more thing, oxyhemoglobin is bright red. Deoxyhemoglobin causes the blood to be dark red. Okay, so keep that in mind. This is what the hemoglobin molecule look like. So here you have an iron here. Hope?
I don't know, working. Yes. It on mute, the mic on mute. Diana? Sir,
Hi.
if you can hear me, sir?
Yeah, I'm hearing you.
Oh, okay. So I wanted to ask a question. Is it true if you don't have enough iron in your blood going to turn yellow?
No, that's not true. If you do not have enough iron in your blood, then you would not be able to carry enough oxygen. Well, first of all, you would not be able to form enough hemoglobin because iron is critical for the formation of hemoglobin. Okay? Now, the hemoglobin carries oxygen. So if you don't have enough iron in your blood, then you would not be able to carry enough oxygen around your body. Okay? Your cells would start to, well, respiration would not be able to occur effectively, and because of that, you would have what you would be oxygen starved. Okay? right? So that's what happens. But women tend to require more iron in the diet than men because of the loss of blood every month. Okay, good. So the hemoglobin has 4 iron groups, 4 iron atoms. So you have 1 iron here, 1 here. You have another one here. Okay, another one here. And when the hemoglobin chain interact with the iron molecule, you get what they call a heme group. Okay, and each of those bind to 1 oxygen, and the oxygen, okay, this will lead to the formation of oxyhemoglobin, okay, causing the blood to be bright red. Okay, yes, Rashid?
So if the cell doesn't have a mitochondria, how come it does get energy?
Well, red blood cells do not have mitochondria, and because of that, remember, the red blood cells do not need energy to transport oxygen. Okay, as long as the blood is flowing, the red blood cells are moving along and the oxygen is being transported. Now, because the red blood cells do not have a mitochondria and they do not have nucleus, they would die every 120 days. Okay, so every 120 days, you have to produce new red blood cells to replace the ones that die because they cannot sustain themselves. Okay, good, very good question. So here we have the equation showing the reaction between hemoglobin, okay, and oxygen, right? The hemoglobin would bind to oxygen to form oxyhemoglobin, okay, HbO2. And it's a reversible reaction. So at the lungs, the hemoglobin would bind to the oxygen to form oxyhemoglobin. When the blood gets to the cells, then the hemoglobin, the oxyhemoglobin would release the oxygen to get back hemoglobin and oxygen again. The oxygen would diffuse out of the blood into the cells for cellular respiration. Okay, so learn that equation.